this is Ava Amson welcoming you to this Bite-Sized Bio webinar, which today is sponsored by Eppendorf. Eppendorf is a biotech company which develops, produces and distributes systems for use in all spheres of life science. The company's broad range of high-quality products and systems is used by researchers worldwide to efficiently carry out their work. Today's presentation is titled Introduction to Liquid Handling for NGS Workflows, and it has been prepared by Elisa Viering and Dr. Tim Schommarts from Eppendorf. Elisa joined Eppendorf in 2021 as a product marketing manager for automated liquid handling and PCR devices. She has previous experience as a global product manager for automated electrophoresis solutions and as a laboratory engineer contributing to quality control, whole genome sequencing, and throughput increase by implementing liquid handling systems. Elisa obtained a master's degree in molecular biotechnology from Goethe University Frankfurt, focusing on developing methods to identify genetic variation in large populations of DNA samples using NGS platforms. Tim joined Eppendorf in 2016 as an application specialist for automated liquid handling, where he acquired a deep understanding of automating lab routines. He is now the global marketing manager for automated liquid handling and plates. Tim has a broad background in diverse workflows, ranging from molecular biology, cell and infection biology, immunology, and biochemistry. He studied molecular life sciences in Germany and Singapore and obtained a PhD in virology from the University of Hamburg, focusing on viral cross-species infection. Unfortunately, due to unforeseen circumstances, Elisa is unable to be with us today, so Tim will take us through the full presentation. As always, we will have a question and answer session afterwards, so please type any questions you have into the questions box which appears on the bottom of your screen, and I'll put them to Tim at the end. So now, over to you, Tim, for the presentation.
Yeah, welcome everyone also from my side, and thank you very much for this kind introduction. As mentioned, I will guide you through this webinar alone today, a little bit different than planned, but yeah, that's the nature of those live events. So we will tackle 4 different topics today. I will give you a very quick introduction to NGS. Then we will move over to tips and tricks parts, which is very valid for a manual handling of NGS libraries. This is really the biggest part of the presentation. I will then present the benefits of automating NGS protocols, and if there is enough time, I will quickly touch a few examples how NGS can be used. But let's jump right in and get started with the introduction to NGS. So the question is a little bit, why was NGS getting more and more popular over the last years, over the last decades? And I think this is very nice depicted here in this graphic coming from the NIH, and it basically boils down to the cost, right? So with the Human Genome Project in 2001, the cost for sequencing a genome was around $10 million, so $10 million for a single genome, and this cost slightly declined, and then there was a sharp decline, and now the costs are falling rapidly. This is actually the cost per genome. So I'm sorry for that. It was $100 million per human genome in the beginning, and now we dropped to around about $1,000 for sequencing a human genome. And of course, this is a major driver why next generation sequencing and different sequencing strategies are now implemented in labs all over the world, and why this is also now slowly but steadily moving into diagnostic purposes as well as even therapeutic purposes. So a short overview of the history of sequencing technologies. In 1977, the foundation was laid with the so-called Sanger method, Sanger sequencing, something which is still valid for today. I will take a look at this in a few slides. Then in 1981, the human mitochondria was sequenced, and then in 1919, the Human Genome Project started. And you can see in between there was the complete cell genome, the complete eukaryotic genome, and then finally in 2001 the Human Genome Project was finished. So it took almost or roughly 11 years to finish the sequencing project. And then in 2005, the first new generation of sequencers came to the market. And this then developed into the second generation of sequencers like the genetic analyzers. And we are now in a phase where we take a look at the human microbiome project. And we are also seeing so-called third generation sequencing. I will talk about this in a minute. So if we compare the three most common sequencing technologies nowadays, we can see that we have the so-called first generation of sequencing, which is classical Sanger sequencing. It's still used today. The advantages is that it is pretty accurate. It has a low cost if you have less than 20 amplicons, and it has a rather fast turnaround time. The disadvantages are that it has very limited capacity. The costs are drastically increasing if you have more than 20 amplicons and you have limited throughput. Then we have the so-called second or next generation sequencing, which is Illumina sequencing, for example. The advantages is that it's easy to apply, the costs are low, you have a pretty high throughput. A few of the disadvantages are that the coverage and mapping is sometimes not ideal. The data interpretation is very challenging, so you need bioinformatics for that, and it's tough to detect structural variants. And in order to tackle that last point, we now have the third generation of sequencing, PacBio, as well as Nanopore. Their coverage and mapping is way better. It allows for de novo assembly, and you can easily detect structural variants. The disadvantage is that it is still not very accurate today. It's very costly, and the library preparation is very expensive. So, but what are the differences? So what is the underlying principle of the sequencing technology? So I think every one of you is probably familiar with Sanger sequencing. So very simplified, DNA is fragmented and is then amplified in the presence of DD-NTPs, which leads to a chain disruption whenever this DD-NTP is incorporated. So that means that at the end, you have basically fragments which with ending with different DD NTPs, and those can be run with an electrophoresis. So they will be sorted by length, and you can detect the DD NTP, and this then gives you sort of a linear read, and it gives you the sequence. And it's still widely used for routine applications, but the sequencing capacities and the speed are very limited. And second generation sequencing, so-called NGS, is then built upon this. So in the beginning, there is still a fragmentation, but now those fragments are attached with adapters. There's an adapter ligation. And instead of having a linear gel, they are attached to an array. So we get a two-dimensional picture. And then we go through cyclic array sequencing, and we can detect all of those different points at the same time during different cycles. And that allows of more than 1 million reads per array. So a massive increase in the number of reads that you get. So in a very simplified way, you start with the library preparation, then you go through clonal amplification, and in the end, you have the actual sequencing. But especially with the second generation of sequencing, so-called NGS, there are a lot of different flavors. So library preparation very much depends on what you want to look at. So there's everything from exome sequencing down to single cell sequencing, clonal amplification. There are two main differences. It can be bridge PCR or emulgent PCR. And then for the sequencing itself, there are several systems out there. Most common is SBS sequencing by synthesis from Illumina, or the ion semiconductor by Life Technology. So these are the most common techniques that are used nowadays. Very quickly touching third generation sequencing, so-called maybe next generation sequencing. So there are two different techniques. One is the PacBio system. This is based on the circularization of rather long fragments, up to 100 kilobases, and with a zero mode waveguide. So that's a nanostructure, and the help of fluorescent dyes. This system can then have very long reads and sequence very long reads. So that's a very elaborate technique. I'm not an expert on that. There are brilliant webinars from Pacific Bioscience, which you can watch if you are more interested. Same holds true for Oxford Nanopore. That's actually a technique that doesn't use any fluorescent dyes. So you can work with even longer fragments, up to 2 omega base pairs attached to a motor protein and to an adapter, and they are then guided through the so-called nanopore via the motor protein. And based on the electrical current readout, which you can see here, you can then calculate back on the base pairing of this sequence. So, but currently, this third generation sequencing, there is no widespread use. It's very specific applications that demand this. It allows for very long reads, but it's way less standardized as compared to second generation sequencing. And that is the reason why during this presentation, I want to focus on the so-called next generation sequencing, second generation sequencing, because it comes in so many different flavors. It can be applied for so many different applications and so many different scientific questions. And the sequencing systems itself, they range from a very small system like the ISIC here. So these are all Illumina systems, up to a very big system. The biggest sequencer from Illumina those days, the NovaSeq. So you can very much have a sequencer depending on your specific sequencing needs. Okay. So that's the background. Now I want to move into tips and tricks, mainly for NGS library preparation. And in order to get started, let's take a look at a very simplified NGS library preparation workflow. So it all starts with the primary sample. It can be blood, it can be saliva, it can be any type of tissue or microorganisms. And you then have to extract the nucleic acids that you want to analyze. Can be DNA, can also be RNA, via nucleic acid extraction. So, and that is a story on its own, so to speak. So if you follow this link here, or if you scan this QR code, you will be directed to a different webinar, which is focused on nucleic acid extraction. It would be too much if we covered this topic here as well, but maybe as a rule of thumb, so make sure that you have proper nucleic acid extraction, that this is an optimized protocol, because also for NGS, there is the rule of garbage in, garbage out. So the better the input, the better your final result. What I can always recommend is a quality control directly after nucleic acid extraction. First, a quantification to determine the exact input to move into your NGS library preparation, and also a qualification, so how good are my extracted nucleic acids. This allows for a good troubleshoot and optimization protocol. And you can adapt the fragmentation if needed. I will come to that in a minute. And then after this QC, we move into the actual NGS library preparation. And this can be split up into 4 major parts. We have fragmentation, we have end repair and A-tailing, followed by adapter ligation, and then we move into the PCR amplification before we continue further to the actual normalization and sequencing. I will guide you through each and every one of those steps. So let's start with the fragmentation. So the idea of the fragmentation is to break down your long nucleic acid strands into fragments of a specific length. So typically we're talking about 150 base pairs. And this can be done with physical measures, for example, sonication or acoustic shearing. So what you get is out of a large nucleic acid strand, you get smaller strands, right? And this is a mix. So you have some strands which have your 150 base pairs, but there are longer and smaller fragments. And typically you want to select for the fragments around 150 base pairs, so-called size selection. And this is the first time where bead handling comes into play. We will talk about beads a lot during this webinar. So these are small magnetic beads which can capture nucleic acids and which are used during the process of NGS library preparation. And for the size selection, the first important thing to remember is that the volume ratio from bead solution to sample volume is absolutely crucial to get the correct size selected. So it's been depicted here. So you have a depiction of the number of fragments on the y-axis, and on the x-axis you have the size of the fragments. And you can see here, depending on the ratio from bead to DNA, so 0.8, 0.65 or 0.5, you select for different fragments of your fragmented nucleic acids. And in order to get a good size selection, make sure to pick the right volume ratio and make sure to have it well mixed, have a homogeneous bead solution, and also that you do not have any evaporation of your sample volume or of your bead volume. So the first important parameter which impacts NGS library preparation is bead handling. And as said, we will come back to the bead handling later during the course of this webinar. Then let's, after the fragmentation, if you have the option, it might make sense to do a quick quality control. So the quantification to ensure that you still have the correct input amount, and of course, a qualification as shown to check if you have the right size distribution. If you see that something is off here at that point, you can really avoid the effort and the costs for the library preparation. So you basically can start optimization before you open a very expensive NGS library preparation kit. It saves you a lot of costs. And this QC helps you to troubleshoot and optimize your protocol. Okay, after the fragmentation, we move into end repair and A-tailing, because what you can see here is all of those fragments. So there are some fragments with blunt ends, there are some fragments with sticky ends, so overhangs at one or both sides. And in order to sequence a library, we need blunt ends for all of them. So what happens here is a so-called end repair. All fragments will be filled up so that you have blunt ends at the end. And there will be so-called A-tailing. There will be an adenine overhang added, and this is then needed later on for the adapter ligation. In a lot of protocols for NGS kits that you can buy those days, the fragmentation, end repair, and A-tailing is combined. So this is a so-called combined step. It's then called tegmentation. And this is usually done enzymatically. So you have your whole, for example, whole genomic DNA, and then with a mix of different enzymes, there are several things happening simultaneously. So you cut this into smaller pieces and you directly do the end repair as well as the attachment of the A overhangs, right? Right, okay. Following this, so either tegmentation or end repair and A-tailing, you need to attach adapters for the sequencing of the DNA fragments. And these adapters are needed to bind your sequences to the flow cells for the actual sequencing, for the bridge amplification, and also for the start point of sequencing. And they can also serve as an identifier for the sample. So imagine them as some sort of barcode. Those adapters are usually 75 base pairs long. So a successful adapter ligation will show a size shift compared to the fragmented DNA. So we said that you were aiming for 150 base pairs fragments. If you then add 2 adapters, you add 2 times 75 base pairs, so you end up with roughly 300 base pairs of a library. And if you do not see this this size shift, or if you see some sort of shoulder, this shows you that something went wrong with the adapter ligation, and you can adjust for that during troubleshooting and optimization. Okay, so, and if we talk about end repair A-tailing adapter ligation, so how is that driven? And this is all driven by different enzymes. And in order for those enzymes to work, you need to ensure that you have the optimal temperature conditions for this. So next to bead handling, another important parameter that impacts the success of NGS library preparation is the temperature incubation. So you have to ensure that your sample reaches the optimal temperature in order for the enzymes to function correctly. This can either be done with a thermomixer or thermoblock of some sort, or for certain steps in a PCR cycler. What is a general rule of thumb and important to know, for example, for the segmentation, if you work with a thermomixer or thermomodule, so you have the heat transfer from the module to the sample, and please use high quality heaters like a thermomodule, so don't go with a water bath. This often gives you some problems besides the contamination risk. And of course, if you are heating up a sample, you have to make sure to use some good sealing, some sort of vapor lock. It can be an oil overlay, but it can also be closed by a foil, because you don't want to lose any of your sample volume during that. Especially for automation, but also for manual handling, I think it's always important to imagine that if you have your samples, for example, here in a plate, and this sample needs to reach 75 degrees, for example, the 75 degrees that your thermoblock or your thermomixer shows you is a temperature that is reached at the heating plate itself, right? So in between, you sometimes have an adapter, and then you have your consumable. So that means that this heat needs to go through several layers to reach your sample. What you want to have at the end is those 75 degrees in your sample, all right? And in order to make sure that this is really happening, it's important that you ensure a tight fit between the module, the adapter, and the consumable. You can imagine that if you have some air gaps in between here, it's a perfect isolator, so you will not reach the temperature in your sample. Use good consumables, which have a good heat transfer, but which are also not melting or shifting or bending. What can also help is to add a few degrees for higher temperature steps. So for example, if you need to reach more than 65 degrees, you can add 2 degrees to the heat. Or you can also add some extra time for those steps. So again, more than 65 degrees, you can add 2 minutes to the incubation time. So this often helps if you're encountering some problems with one of your incubation steps. If we then, after end repair and ligation, and we move into PCR amplification, this is to increase the amount of adapter ligated libraries. So with primers, as you can see here, that you just have more material in the end to sequence. Of course, it's important to always use a PCR cycler to do this. It sounds very trivial, but I have seen different approaches to do this. So it's recommended to use a high quality PCR cycler, because you don't want to risk your experiment because your PCR here at the last step is failing, right? Okay, so that guided us through the whole NGS library preparation protocol. And there is one final quality control step, and that is the quality control step that you should never skip. it's really crucial to do a quality control here in order to move on, to move into normalization and to move to sequencing. And it's also important that you do both quantification and qualification because you don't want to have the costs for sequencing if you already know here at this step that you ended up with a poor quality library. For quantification, this is usually done by UV vis or fluorescence, for example, a qubit. If you want to be very precise, you can use qPCR. That's actually the gold standard, but obviously also a bit more work. And for qualification, some sort of electrophoresis. It can be a gel, although this is very tedious to handle. So most of the labs are nowadays using either bioanalyzer or tape station or a similar system for automated electrophoresis. So, but how to identify a good or a bad library? We are coming back. So this is a quality control. So and these are tape station traces. And thank you to Agilent Technologies for providing us this pictures here. So what you can see here on the x-axis is the size, and on the y-axis is the intensity, so the yield. And you can see here, so this is a very good looking library. So we have two intrinsic markers, lower and upper, and then we have our library signal here at around 300 base pairs. It's a very good size distribution. There are no adapted dimers and no bubble peaks. So this is a quote unquote perfect library to continue and to sequence. If we take a look at the bad library, so here we have a good size distribution, but we have no adapted dimers, but we have this so-called bubble peak here. So this is this shoulder. These are usually unspecific longer fragments. It can be that there was too much template input or too many PCR cycles. It's not perfect, but it can still be sequenced. There is, however, the risk for a wrong estimation of your library concentration, which can affect your sequencing results. But this is sets more like a yellow card, so this is not a big problem. A bigger problem looks like this. This is a big amount of so-called primer dimers. So you can see here the size distribution of the library is okay. Also, the yield is okay. But you have this very sharp peak at around 150 base pairs. These are so-called adapter dimers. And the problem is that they don't only like to bind to your beads, they also like to bind to the flow cell, and this will negatively affect your sequencing quality. Possible causes are that your bead to sample volume ratios of that your sample volume is lower than expected, or your bead volume is too high, or that your bead mixing is not correct. This all causes insufficient size selection. And as you can see, it all comes down to bead handling. So I will quickly jump back to the bead handling, and you can see here two types of bead handling. So these are insufficiently mixed beads, so you can see two layers. And this is a good mixing or perfect mixing. So use high quality mixers with a big radius, thermo mixers, or if you see that you have this type of bead mixing, implement some tip mixing. The second important thing when it comes to beads for the handling itself, so that you don't have a carryover of beads, is to use strong magnets with a tight fit, no matter if it is a magnet for tubes, as you can see here, where the magnet is coming from the side, or if this is a plate magnet, as you can see here, where you have a ring magnet. So if you have an improper fit, you will always have floating magnetic beads, which will carry over and cause problems in the next steps. If you have a tight fit between your consumable and the magnet, you have a good separation, which then allows for easy pipetting and a good and clean process. All right. Okay. One last thing when it comes to the beads is the bead drying. At some point of the protocol, you have to dry the beads to get rid of remaining washing liquid, so ethanol. So there are basically three scenarios. One scenario is that it is too wet, so it's not dried properly. There is still a lot of residual ethanol. This ethanol will interfere with follow-up steps. So ethanol interferes with a lot of enzymes, and it will cause problems with the next step. The other option is that you over-dried your beads. The beads are then too dry. They will be brittle. And the problem is that it's very hard to elute DNA from those over-dried beads. And here in the center, you can, of course, see the perfect scenario, very little residual ethanol, but the beads are looking good and they are not over-dried. So in order to ensure this optimal drying, make sure to completely empty your wells, maybe use different tip sizes to do that. Determine your best dry time in an optimization step, and under no circumstances support the drying with heat. This is something that is definitely not working well. All right, so we talked about bead handling, we talked about incubation and temperature. But there is one factor which is also has big impact on the success of your NGS experiment, in fact, of every experiment, and this is the quality of pipetting and liquid handling. So very quickly, you're probably all familiar with that for the pipetting cycle. So if we talk about manual handling, then you have the aspiration of your liquid. If you want to dispense it again, you have first the dispense step. There's always a little residual volume left, and then you do the so-called blowout that you get your tip completely empty. If we are in an automated scenario, it's more or less the same. The only difference is that in between you have this so-called trailing air gap that is generated. So if a system is moving around, that you have no dripping. Yeah. So what is important when it comes to pipetting, besides a good pipetting technique, I think is the use of the proper tip. If we talk about automation, I always prefer transparent tips instead of conductive black tips, because you can see what is happening in a tip. So for example, here you can see that there is still a lot of residual volume left in your tip. So probably you don't get enough enzyme or reagent into your reaction. That is problematic. And that is a tip which is looking good. So if you are in the optimization protocol, take a careful look of any residual liquids which might stay in the tips. You probably also have spotted a problem here if we talk about NGS. So if we talk about tips, always use filter tips. Yeah, this filter is an extra protection, especially for aerosols that are generated. Most NGS protocols include an amplification step. That means if you have a contamination of your pipette, this contamination will be carried over into your actual sample. And let me tell you a story from my time in the lab. There was a lab which was working with adenovirus, and actually all of their pipettes were in one room were contaminated with a residual adenoviral DNA. So that means when they had to run very delicate experiments to detect adenoviral DNA, they needed to do this in a completely different lab with a completely different set of pipettes, because this very little traces of DNA at the pipette were enough to spoil their experiments. Yeah? So make sure to use filter tips for an extra layer of protection. If we talk about automated dispensing, we also can talk about two different modes. One mode is the so-called pipetting mode, where you take up a certain volume and then release the same amount of volume. This gives you high accuracy, but it can be rather slow. But the blowout allows you to really eject remaining liquid. And of course, you have to, you can change the tip between each aspiration step. In contrast, we have the so-called multi-dispense mode or sequential dispensing. It's faster but less accurate than pipetting mode. That means you take up a multitude of your dispensing liquid, and then you dispense this stepwise. So first dispense, second dispense, third dispense, and then a little bit of residual liquid, which either goes back to the source or is discarded. And of course, here a tip change between these different dispensing steps is not possible. When to use which pipetting technique? I would always use the pipette mode for samples or critical enzymes, and multi-dispense for buffer. Of course, this only applies if you use automated dispensing or electronic pipettes. If you work with mechanical pipettes, you always fall back to the actual pipetting mode unless you are using a repeater system. All right, so that is the last parameter which is important for the success of NGS library preparation, and that is the pipetting itself. Okay, so I talked a lot about tips and tricks for manual process, but I mentioned a couple of times this is also interesting if you automate your NGS protocol. And now very quickly, I want to tell you why it might be beneficial for you to think about automating NGS protocols. So coming back to our beloved depiction of the workflow. So the question is, why can you automate those protocols at all? And the reason is that, especially here, the central part, nucleic acid extraction, library preparation, normalization, as well as the quality controls, they are usually very standardized and mainly based on kits. And those are processes that can easily be automated. Okay, so it can be automated. So the question is, but why should I do that? Why should I automate it? The first and very obvious answer is throughput. So if you implement automation, it's much easier for you to increase your throughput. So as a person, as a human, you are maybe capable of handling 8, 16, 24 samples, but there is a limit if it comes to working concentrated. But for a robot, they can easily handle 96 or even more samples in a single run without getting tired, without running into mistakes. But you might ask yourself, okay, that makes sense. But in most NGS labs, if we talk about non-core labs, there are not hundreds of samples every day that need to be processed. So if I have less samples, does it still make sense for me to automate my workflow? And the answer is, of course, yes. Otherwise, I would not do this motivation here. And the reason is just based on the workload that comes with this different steps. So we have about 500 pipetting steps, 2 hours hands-on time for nucleic acid extraction, 2,000 pipetting steps, 5 hours hands-on time. NGS library preparation can be easily more than 5 hours. And then also 100+ steps for the QC and the normalization. And this is tedious. This is very demanding for the operators. You need to stay focused and concentrated for a long time. And of course, if we talk about 2,000 pipetting steps or more, it's also very error prone. So it's very likely that at some point you make a mistake, you forget something, you put the wrong volume to your pipette. It does not need to happen, but it can happen very easily. And so that means you need to stay razor focused on your experiment, on your samples, on your pipetting. And if you give these tasks to a robot and the robot does the handling, of course, you are free to concentrate on other tasks, which is usually data analysis. So the biggest advantage for automation, or one of the biggest advantages, is that it frees time. It frees time for other things to do. Right. So in a nutshell, when does it make sense to automate? One big component we talked about this is the throughput. So if you want to increase your throughput, also if you want to have increased accuracy and precision, and I'm not talking about a single experiment, but over the course of several experiments, a robot will usually be more accurate and precise, as different operators during a manual process. Of course, a automated system is basically human error free, unless there are some input errors. It gives you additional data integrity, and it helps with the documentation. So you don't have to write a lab notebook, because the system is keeping track of what is happening, and it adds an extra layer of sample and user protection. But for me, the most important thing is reproducibility and standardization. So if you run a protocol that needs to be or that needs to run several times in your laboratory, and if you do this in an automated fashion, you can be sure that every time the performance is the same, every time the handling is the same, and also that you get roughly the same output from your experiments. And this is allow me one product slide. So this is really the only product slide that I'm going to show you here. So for NGS library prep, Eppendorf is offering several automation systems, our so-called epMotion systems. For systems, we can take a look here. So we have two smaller systems, epMotion 96 and epMotion 5070. So this is so-called beginner automation, or for the 96, it's a semi-automated system. So these are so-called, I would call them helper systems, because they cannot automate the whole workflow, but they are perfectly capable of supporting you with the most tedious tasks, like bead purification, or maybe with a qPCR setup, or filling of plates, something like that. And then we also have a full solution. These are the larger systems, 5073 and 5075. These are two different sizes. So this is for low to medium throughput of NGS library preparation. And this is for medium to high throughput of NGS library preparation. So this is really a full solution for this. And the good thing is that these are all open systems, so they can not only be used for NGS library preparation, but as said, also for your nucleic acid extraction or for your normalization, as well as other tasks in the lab. All right. Okay, and since we have a little more time than expected, I would move on to some examples for NGS. I only brought 2 examples, so don't be afraid. It's not going to be too much here. One is the often talked about microbiome project, so the so-called human microbiome project. It's a very interesting field of research those days. So as you all know, the human is host to a myriad of different microbes, either on your skin or in your gut or in every part of your body. And those microbes very much shape different, let's say, physical features like digestion, your susceptibility to certain diseases. So over the last decades, it became more and more en vogue and more interesting to look at the human microbiome. And this can be easily done with next generation sequencing. So there is one option which is 16S ribosomal RNA gene profiling. So this allows to detect for bacteria and archaea. So this is a very specific and pinpointed approach, but this can also be used for so-called shotgun approaches, so where you just sequence the whole, for example, gut microbiota, and you can then map metagenomics on the one side, so what kind of microbes are present to detect functional pathways, but you can also do meta-transcriptomes, where you look at the whole transcriptome of a set or a subset of microbes, which allows you to see which pathways of those microbes are active, and this allows you to track back and to identify things that are caused by maybe changes in the microbiome, or if you have overrepresentation of a certain subsets of microbes in a certain area of your body. The second sequencing application is something that we are all very familiar with, or that we heard of over the last days, is so-called virus surveillance. That's the example of coronavirus Omicron. So this is one of the first articles when Omicron appeared from Al Jazeera. So this was in late 2021. And you can see here that it was pretty much unknown how Omicron looked like. So you know that there were 32 mutations, but you didn't know what the virus was doing. So how do you identify those mutations? So this is a depiction of the coronavirus this is the proteome, so the coronavirus proteins and how they are encoded. And you can see here the Delta variant of corona with the different mutations. So these are those lines. The lines are the mutations. If there is no indication here, then this is the so-called wild type virus, and you can see that the Delta virus has roughly 10 mutations in their proteins, which cause a different behavior, which make it more aggressive, so to speak. And then if we look at Omicron, you can see that there are a lot of mutations, so a total of 32. And it's important to identify those mutations, because those mutations allow you to predict how a virus will behave, or it even helps you to tailor-make a vaccine, or if we look into the future, maybe active medication for this virus. And if you can see this widespread of mutations here for the Omicron virus, this has been detected with sequencing, with next generation sequencing, and the different virus clades all over the world are still monitored by this virus surveillance technique to ideally spot newly arising virus mutants before they spread around the world to react quicker than the virus can do. Yeah, and with this, I want to finish, and I thank all of you for sticking with me here to the end of this talk. I know it's been heavy stuff, but I hope it gave you an insight on NGS, and especially how you can improve your NGS workflow, and maybe you can take 1 or 2 tips or tricks and implement it into your own workflow. And now I'm very happy to take a few questions.
Thank you, Tim. That was a great presentation. So we've got a few questions from the audience already. If anyone else has a question, you can leave that in the Q&A box that's at the bottom of your screen. So the first question I've got for you is, are there any alternative methods to NGS?
Yeah, very, very good question, and actually not an easy answer to that. That very much depends on the actual question that you are pursuing. So if it's really de novo sequencing of an unknown sequence, an unknown genome, there are hardly any ways around NGS. So to a limited extent, you can replace it by Sanger sequencing. If we look at a different approach, for example, where you do so-called target sequencing, where you look at specific mutations at specific genes, there is a limited chance to replace this with a multiplex PCR. If it's transcriptome, to a limited extent, you can replace NGS with qPCR, so real-time PCR. And you probably don't have the setup for this, but sometimes it might also make sense to do proteomics. So instead of looking at the genome or the transcriptome, to look at the proteome. But as you see, there's a lot of different methods, and NGS can do all of it. So it's tough to find a really good alternative.
Yeah. And the next question is, how do I know if the kit that I'm using can run on a liquid handler?
Yeah, also very good question. I thought about that question when it popped up in the chat during the course of the webinar. So I think that the best way is to check at manufacturer websites. So both NGS kit manufacturers like Illumina, but also liquid handler manufacturers like Eppendorf usually have an overview of which methods are available on what system. They then still need to be modified for your specific needs and purposes. We at Eppendorf, we have a special service called application implementation, where you get a visit from one of our specialists, and then they are setting up a specific method on your automation system, Eppendorf automation system. And that's probably the best way to get started. But I will try to use the chat and post a link here. I don't know if everyone can see this then. So this is a...
Yeah, if you set it to everyone, then everyone can see it.
Let me see if that works. Okay, here we go. That's the method overview from Eppendorf, so you can browse it and you can see what's available on our automation system.
Thanks, that's really useful. So the next question is qubit or a picogreen cost effective to quantify a library composed by multiple plates, 384 wells? Are there any alternative?
Yeah, I have to think about it a second. So it's a very, very specific question. So from the two options that are described there, I would say that probably picogreen is the better option, the more cost efficient option if combined with a plate reader. There is one probably more cost effective method, which is a nanodrop. But you will surely not want to go through 384 samples using a nanodrop, so that's very tedious. And the last option that I could think of is would to do qPCR. That's actually very good for quantification, but it's a bit more work to get it started. Or to directly use the quantification option on your bioanalyzer or tape station. It is not ideal, but it can be used. The one important thing that I want to add here is that if you have decided on a certain QC workflow, stick to it. So don't change it between experiments, because it gets very hard to compare the quality of your libraries then in between different isolations.
Thank you. I'm sure that was really helpful. Next question is about sample preparation. When you're using the enzymatic tagmentation, do you still need to fragment DNA, for example, using ultrasonic waves?
Yeah, very good question as well. It's a number of really great questions coming in here today. So if the protocol has a tagmentation included, so the sort of enzymatic break off of longer fragments, there is no additional fragmentation needed. So in fact, it might even be a problem if you fragment it first and then digest it again with an enzyme. So usually the kit will tell you. So I think most of the Illumina kits out there, which are pretty common, but also other kits, for example, from Kappa Biosystems from Roche, they use the tagmentation. It has the advantage that you do not need extra instrumentation like a Covaris. On the other hand, you are a little bit less flexible when it comes to the fragmentation step, because it's already included into let's say or combined with two more steps. Yeah, but don't do both. Don't do tagmentation and then fragmentation as well.
Thank you. The next question is, what are the most common causes for DNA loss during library prep?
That again is an interesting question. It depends a bit on the handling, I would guess. So it's of course a bit of hand waving for me here, because protocols are very different. I think the biggest risk to lose DNA, so to have a reduced yield, is the bead handling. And for example, during your washing steps, if you don't have a strong magnet, for example, you will aspirate beads with your washing solution. So you will constantly lose beads, and your target DNA is still attached to those beads. So, and if you do this multiple times over the course of the NGS library preparation, I would guess that this is probably the most common cause for a low yield. So check your pipetting, check if you see that you have this kind of brownish rust-like traces while pipetting. That means you're losing beads. And then the second option is drying. That can be problematic, as mentioned. So if the beads are too dry, if they look brittle and cracked, and it's a color shift, it's a little bit of an experience thing, then the illusion the illusion efficiency is drastically reduced, and there you lose a lot of DNA to the beads. Yeah, that's I think are the most common causes, but it can have different reasons as well.
Thanks. And then the last question. Oh, no, more came in. We've got, let me just have a quick check. I'm going to leave the question about data analysis for the end because I want to combine it with another question. So I'm just going to jump ahead a little bit. Can NAP and NGS library prep be automated sequentially without interruption, or do they need to be split as two independent tasks?
So on the epMotion, system, it cannot be combined into one, let's say, very long protocol. This is due to the size of the systems and the capacity of the deck. I would guess that if you have a much larger liquid handling system, there might be an option to combine it. However, I would not suggest you do it because if you do your nucleic acid purification, you want to do a quality control afterwards, right? Mm-hmm. So imagine something goes wrong. There is very little DNA or RNA extracted, especially RNA is tricky, and then you directly move on. You will move with this poor quality input material into your NGS protocol. In fact, I have seen a lot of labs that are even splitting pre and post PCR processing. If you have everything on one device, it works. It depends a bit, but if you have very delicate critical protocols, it can even be a problem to have your amplified sample on the same system in the same space as your non-amplified samples. So there's no easy answer to that, but I would definitely not suggest to combine it on a single system.
And then a quick question is, what is the minimum volume that can be handled by your liquid handling workstations?
Yeah, we're diving into tech specs here. Yeah, the minimum volume is 0.2 microliters, but that volume can only be effectively handled if we have so-called contact dispensing. So if we dispense directly into another liquid with the so-called free jet dispensing, we can go down to, I think, realistically 1 microliter. So the epMotion is covering a volume range between 0.2 microliter and 1,000 microliter. And that holds true for all the systems.
That's a good range. And then to end on this last question is, could you comment on some data analysis tools? And I wanted to expand that a little bit by also asking you if you could recommend a specific quality control method or tool.
So maybe I start with the quality control because that comes first, and that's also easier to answer for me. The quality control, I would personally suggest a combination of the qubit and this is for the for the quantification, so it's important that you do both quantification and qualification of your libraries. So for the quantification, I would suggest a qubit. It's a very good system. A picogreen is also an option, as mentioned before. I would say that for quantification, the gold standard is qPCR. So there are so-called library quantification kits out there, which are qPCR based. This is if you are really encountering some issues that you over or underestimate your yield, then this might be helpful. For the qualification, so the quality of your libraries, as shown, I think the best option is to go with a tape station, which is an electrophoresis device. It very clearly shows you if you have quote unquote good looking library or if you see any problems. If we move to data analysis, the situation is looking a bit different. Honestly, I'm not a bioinformatic expert. There are different, let's say, providers out there. I think that the most common one is Illumina base space, I think it is called, where you can have a subscription for this. This gives you a number of tools to do interpretation on your own of your data. Then, of course, I think that a lot of core labs, they work with their own bioinformatics department. So that was actually the way that I was doing NGS. I was doing the library prep, and then it went into a sequencing facility, and I got the results back. That's probably the most common way. There is maybe one startup company that might be interesting for some of you. They are called Big Omics, a new starter, so it might make sense to take a look at them. They have a special option for academics, and they are specialized on transcriptome analysis. So if you do this, it might make sense to take a quick look at them. They're called Big Omics, but there are sort of plenty fish in the sea and a lot of different data analysis tools and options.
Yeah, lots to choose from. Yeah. So thank you very much. That brings us to the end of the seminar today. So thank you, Tim, for a very illuminating presentation and discussion. And thanks to Elisa for helping to prepare this presentation. And of course, thanks to our sponsor, Eppendorf. And finally, thanks to you, the audience, for taking the time to attend and listen in today. If you would like to listen to this presentation again, it will be available on demand soon. So until next time, good luck in your research and goodbye from all of us at Eppendorf and Bite Size Bio.